Okay. Yesterday we were covering the mechanisms of insulin secretion, and just want to summarize here. So essentially, you have glucose that binds to these GLUT2 transporters, is taken up, metabolized via glycolysis. This produces ATP. This ATP triggers mobilization or depolarization of the membrane via potassium channels. You then get influx of calcium. This calcium then triggers those endopeptidases. You get the first wave release, right? So first wave release, first phase, which is 5% roughly. Then with the active endopeptidases, you get packaging of proinsulin and then processing into insulin and to produce your second wave, okay? And then in this ATP, remember, we close those potassium channels, depolarize the membrane, and promote vesicle movement via that kinesin. Okay, so let's talk now about a little bit how this is regulated. So insulin production and release are clearly very important for a variety of metabolic features within the body. So this requires a very close monitoring and control of the process. So it means that it becomes highly regulated at a variety of steps. And mutations/defects in any of these can lead to either metabolic disorders, obesity, or in type 2 diabetes. So in addition to glucose, insulin secretion is also regulated by the autonomic nervous system. Okay, so you have neuronal inputs coming in. It's also regulated by other pancreatic hormones, which we'll cover towards the end. It's regulated by intestinal hormones. And it's regulated by the levels of amino acids and fatty acids in the body, which obviously is also directly related to diet and exercise. So how does the autonomic nervous system work in terms of regulating insulin secretion? Well, you have release of noradrenaline onto the beta cells. These act on beta-2 and alpha-2 adrenergic receptors. And then beta-adrenergic stimulation augments insulin secretion, whereas alpha-adrenergic stimulation inhibits it. In addition to the sympathetic nervous system, you also have parasympathetic nervous system inputs. This comes in the form of acetylcholine, which increases insulin secretion via M3 muscarinic receptors, and acetylcholine can increase calcium via the PLC IP3 pathway, and a novel pathway which is directly linked to a protein called beta-arrestin. Finally, the polypeptide galanine, which is also found in some autonomic nervous innervate the islets, and can also inhibit insulin secretion. And almost all of these are very important and of interest to biotech companies for developing novel compounds and drugs, many of them within China, trying to find novel compounds to treat all of these to control diabetes. So in addition, we also have, in addition to the nervous system, you also have control of insulin secretion by pancreatic hormones. So glucagon can stimulate insulin secretion, and somatostatin inhibits insulin secretion. So these are two pancreatic produced hormones that are not directly related to that are related by other cells, if you remember, but still regulate insulin secretion. In addition, you also can have control of intestinal hormones, and these are called incretins. So these hormones, one big example is the glucagon-like polypeptide GLP-1, and its related glucose-dependent insulinotropic polypeptide, or also commonly known as GIP. Finally, there's cholecystokinin, which potentiates the insulin-stimulating effects of amino acids. Now, GLP-1 recently was approved, had a very big clinical trial, and it showed to be incredibly effective at reducing body weight, and was just recently approved by in the U.S. for treatment. So another important drug target for targeting obesity. So this kind of summarizes all of those alternative points of control of insulin that I mentioned to you. You've got the somatostatin galanine alpha adrenergic. You've got your glucagon receptor. You've got your beta adrenergic and alpha adrenergic receptors. You've got your CCK acetylcholine inputs. All of them converging on impacting, right, this secretory granules. Obviously, this is the initial pathway we talked about via glucose. And this is the amino acids, in this case is showing leucine, how they can input by impacting glycolysis and the citric acid cycle, again, on the ATP production side. Okay. So once we have insulin released and secreted, how does it actually work? What is the mechanism of action of insulin? So this is a canonical receptor tyrosine kinase. It's the insulin receptor. And again, one of my favorite exam questions. It's made up of 2 alpha chains and 2 beta chains that dimerize. And if you remember from our earlier lectures, what happens is that the key points of these receptors is once they dimerize and insulin binds these cysteine-rich domains, you get cross-phosphorylation of, in this case, because it's a tyrosine receptor, of tyrosine amino acids on the tail. This then leads to recruitment of a variety of scaffolding proteins for downstream signaling. And then this is depicted here. So you get recruitment in the case of insulin receptor of this SHC kinase, and then this goes on to activate the SAS pathway and eventually something called IRS. These will then go on to impact PI3 kinase, mTOR, P70, CSK3. All of these, if you read the literature on diabetes and insulin, are key components for signal transduction of insulin. Now, what are the consequences of these? You get, in one case, activation of GLUT transporters, so more sent to the surface. You get increase of glucose storage and oxidation. You get an increase in protein synthesis and a decrease in proteolysis. You get an increase in triglyceride synthesis and a decrease in lipolysis and lipid oxidation. And then eventually you get gene expression and growth, like we've seen for many other hormones. So if I were to ask you to write an essay about insulin receptor, I would expect you at least to understand these bottom ones here, and these ones in yellow, if you will. Okay, because those are sort of the physiological outcomes of the receptor activation. I wouldn't expect you to remember all these little signaling downstream pathways, but generally I would like you to know the physiological impact of insulin signaling. This is summarized here. So you have increase of uptake of glucose uptake in most cells via a alteration of those GLUT transporters, increase of glucose use and storage in part by the upregulation of glycolysis, increase in glycogen synthesis, which is your glycogen glycogenesis, increase in protein synthesis, increase in fat triglyceride synthesis, and increase in gene expression and growth. You have typically a decrease in gluconeogenesis, a decrease in glycogenolysis, and a decrease in proteolysis, with a decrease in lipolysis and lipid oxidation. This generally means you have an acute, the stimulus for secretion, is the acute increase in blood glucose, with the main effect a decrease in blood glucose following, okay? Once you release insulin. Now, where are the tissues? They're insulin's major site of action. The liver is a major one. Adipose and the fat is another major one. And obviously, muscle cells are also an important target and site of insulin action. And typically, you can see here where in green what it's actually doing in those cells. So in the muscle cells, obviously, it's going to be driving carbohydrate metabolism, but not necessarily in the liver, whereas the liver, it's going to be more glycogen synthesis and lipogenesis. And like adipose, the major one is reducing lipolysis in general, okay? What about glucagon? So we've talked about glucagon, briefly mentioned it earlier. So the glucagon gene looks like this, and it also is one of these that can be processed into other hormones. In pancreatic islet alpha cells, which we've talked about, we discussed when we were talking about different cell types, is where it gets processed. This gene gets processed into glucagon, okay? And in L cells is where the same gene leads to combination for the production of GLP-1. You don't need to know any of the others. I just would like you to focus on pancreatic islet alpha cells and glucagon and understand that it's the same gene as GLP-1, which is produced in the intestine L cells. What does glucagon do? It acts primarily in the liver to antagonize the effect of insulin. It's also a G protein coupled receptor and does this via GS. It leads to glycogenolytic, gluconeogenic, lipolytic, and ketogenic activity. In the muscle, interestingly, it does not cause glycogenolysis, but increases gluconeogenesis from available amino acids in the liver. And in very high concentration, it accelerates lipolysis in adipose tissue and proteolysis in muscle. And this is one of the attractive features for being able to modulate fat. The stimulus for secretion is typically hypoglycemia, so blood glucose below a certain level. Also, exercise, stress, and high protein meals. This was one of the logics, maybe. I don't know if this was very fashionable in China, but for many, many years, and many people still argue that high protein diets can help control weight. And I know in Western countries, this was a very popular diet a few years ago. And part of it was through this understanding of how GLP-1 worked. Generally, the main effect of GLP-1 is to increase blood glucose. So here's a summary of the role of glucagon, the glucagon receptor, at least in the liver. And again, it binds to this G protein coupled receptor, increased pKa, alters gene expression, and you get major modification of your metabolism. So you can think about insulin and glucagon as an important ratio. So in the fed state, insulin's going to dominate. And then in the fasting state, glucagon will dominate, or also in the exercise state. So what about some other islet cell hormones that we should be aware of? Somatostatin is an important one. It's synthesized in the pancreatic delta cells and then the D cells of the GI tract. And obviously, we've covered it in the hypothalamus and in other sites in the CNS, but importantly, it is made in the pancreas. It inhibits secretion of multiple hormones, including growth hormone, insulin, glucagon, gastrin, and TSH. So it's a major modulator and also a major attractive target for drug companies because it has such a broad effect. It can inhibit basal insulin secretion. However, the challenge is, one, the receptors are found many places, so targeting it from a drug standpoint is difficult. And also the blood flow from the center of each islet. Remember, we talked about the blood coming up, going through the pancreas and then flowing down the mountain, right? From the center of each islet where the beta cells to the periphery of the islet where the delta cells are, it minimizes the effect of somatostatin on the islet from which it is secreted, right? Because beta cells are upstream of this flow of blood and somatostatin is then secreted downstream, it has a harder time impacting insulin of the beta cells upstream. So for diabetes, it's not necessarily an ideal target, but for other indications, somatostatin is of interest. We also discussed briefly pancreatic polypeptide. Human PP is a linear polypeptide containing about 36 amino acids. It is produced by these F cells. Its secretion is under cholinergic control and is increased by a meal containing protein and by fasting, exercise, and acute glycemia. Pancreatic polypeptide slows down absorption of food in humans and may smooth out the peaks and valleys of absorption. The exact physiological function is still to be determined and it is of interest to understand. So briefly, we've talked about the gross anatomy of the pancreas. Remember, it's got that hook shape or like a cane. It got that blood vessel flowing through the middle of it and then seeps out to the other islets. It's made up of a variety of cell types. Its major hormones is insulin secretion. We've also talked about peptide hormone diversity and how that has been achieved. We've talked about mechanisms of glucose-induced insulin secretion, regulation of insulin secretion, and mechanism of action of insulin. And then we've talked a bit about the physiology and biochemistry of glucagon and other cell hormones. So briefly now, I just want to give